Today I want to talk about the first key processing component of a neural network, which is the neuron. Now, one of the things we're really concerned about is, okay, neurons are these key processing components. How do they do it? And one way to think about what neurons are doing is they're sort of detectors, and that each neuron is detecting some set of conditions, like a smoke detector detects smoke. For example, suppose you have a setup like you have here, and basically you have a feature or a neuron that or a feature that basically indicates striped, orange, sharp teeth, furry, and roars. And they all send information to that top node or neuron, which basically serves as a detector to see whether those features are on or off or not. So you might think of that top node as essentially like a lion or a tiger detector. Now, if you want to understand how these neurons behave, it's useful to sort of think about, well, what does an abstract detector look like, and how do those components of an abstract detector correspond to a neuron? So on the one side of the screen, you have the detector, and you have those red lines that are the inputs. They're coming into these blue lines, which are then taking information into that central triangle, which is the integrator, which adds up all the signals that are coming in. And then based on the integration, it then sends information or activation on that red output line. Now, if you then look on the other side, on the red side, labeled neuron, basically what you have is you have these red lines that are coming in, which are the axons from other neurons. And they then hit synapses. These synapses are on the dendrites. And basically, the axons send neurotransmitters across the synapses, which then basically causes a current to flow on the other side in the dendrites. That information then moves to the cell body, where the cell body essentially integrates the information that comes from the dendrites. And then the cell body, after it sort of integrates that information, decides whether or not to fire and send a signal down the axon. Now, so basically in this detector model then, is each neuron detects some set of conditions. And what makes this interesting and what makes it powerful in terms of a neural network and in terms of the brain is that neurons feed on each other's outputs. So you end up with these layers of ever more complicated detectors. Things can get very complex in terms of content. You know, we're talking about stories, language, pictures of animals, cats, things like that. But each neuron is still carrying out a basic detector function. It's just basically saying, I have a particular signal I'm looking for, and do I get that signal? And so you can think of, like in sensory or visual systems, the brain starts out, okay, we have pixels or dots on the retina, then we detect bars of lights. Then you have detectors that can take the bars of light and create edges, and then at several layers higher, you now have cells that can start to detect tigers from all the information about the edges. In the motor system, you might sort of the motor system will detect appropriate conditions or places to move the hand. You have abstract internal actions or abstract internal semantic representations, and other kinds of representations as well in other parts of the brain. Now, one sort of interesting way to think about what these simple detectors are doing when you put them together is a model called Pandemonium that was actually developed by a guy named Oliver Selfridge. And this is sort of a conceptual model. And the way you might think about it is on the left side of the screen you have an R. And that R basically is out in the world, and then you have an image daemon, which basically represents that R. And then with that image, so the image daemon represents the R, and then you have a whole set of feature daemons, each one of which is looking for a different feature. So the top feature is looking for a vertical line, Next feature daemon is looking for a horizontal line. Then you have lines at angles, circles, semicircles, etc. And then each of those feature daemons, basically, if they see something, they're going to send a signal. And then you have these cognitive daemons that are sitting there looking for information from the feature daemons. And they're looking to see if they're getting the right kind and amount of information from feature daemons that will allow them to identify a letter. So you have the feature daemon A that's basically sitting there and going, well, I get information about a vertical line, and I get some information about some semicircles. And then you have other cognitive daemons that are also getting information. But as you go down to the bottom, the cognitive daemon that's actually getting the most consistent information is the R daemon. And it's getting information about a vertical line, a semicircular line, and then a line at a 45-degree angle. And then that cognitive daemon, because it's getting all this information from the feature daemons, is basically going to send the strongest signal to the decision daemon, and the decision daemon is going to say, I see an R. Now, sort of a more interesting or sort of a more interesting description of it or picture of it is actually the original from Oliver Selfridge. Here you sort of see the image daemons, and they're basically sending information to the feature daemons. The feature daemons, if they see something, they start yelling and sending information to the cognitive daemons. The cognitive daemons start yelling depending upon how much information they get. And then the decision daemon is basically sitting there listening to figure out which of the cognitive daemons is basically yelling the loudest. So you can think of sort of a detector system as operating that way. So in this pandemonium example, each neuron has a simple job, but together you put them together with layers of more and more complicated detectors, and you basically go from being able to identify just lines or semicircles or things like that to the point where you can actually identify letters. And you can obviously think of further layers of these daemons where you take individual letters, and now you can sort of learn to, or you'll basically start developing a system where you can recognize actual words. So this is a simple example, but it does raise the question of what kinds of detectors do you need for language, face recognition, creativity, etc. So here's a simple idea of how you might sort of think about the brain as sort of this hierarchy of systems or hierarchy of feature detectors. And how would we simulate this? Well, one way you can do it is you can take neural activity, and later we're going to talk about learning, and characterize them in terms of mathematical equations. We can then use these equations to specify the behavior of artificial neurons. And once we have that set of equations for each individual neuron, we can then take those and put them together into networks of neurons. So let's start with a real neuron. And what are the basic properties of a real neuron? And let's review those basic properties and then see if maybe we can abstract away from that and come up with sort of a set of equations that will allow us to characterize, mathematically characterize a neuron in such a way that it's useful for developing a neural network model. So the basic property of a neuron is it's a cell. It's got body, it's got a membrane, nucleus, DNA, RNA, proteins, etc. Now, one thing for our purposes is maybe some of that stuff doesn't really matter in terms of trying to figure out how a neuron works as a detector. So maybe for some purposes we don't really care about the fact that it has DNA or RNA. Maybe there are other things that are more important. So some of the things that are probably the most important are, well, you can think of neuron as basically containing a lot of different kinds of ions or charged particles. They're present both inside the neuron, but also outside the neuron. And the ones that are most important are going to be sodium, chloride, potassium, and calcium. And if you think about it, sodium and chloride, for example, create salt. In addition, with the cell body, you have channels that allow ions, such as sodium, to pass through. And the channels can be open or closed, selective permeability. So, for example, one of the things that neurotransmitters do is they open or close channels in the cell body that allow ions to move in and out of the cell body. Now, when a neuron is at rest, you have a greater concentration of negative ions inside the neuron versus outside. This difference in charge inside versus outside the neuron is called the membrane potential. And this membrane potential is really critical in understanding the role of the neuron and the ability of the neuron to basically serve as a detector that can send signals. So here's sort of a abstract diagram of the neuron and its ions. So the simplest is probably sort of the aqua one called K+, which is potassium. That's a positive ion, but potassium is continually leaking out of the cell body. And because it's a positive ion and it's moving out of the cell, it reduces the membrane potential or sends the membrane potential of the cell to be negative. In addition, then, you have the chloride ions, the Cl-, and this is serves as an inhibitory or synaptic input. And chloride, basically, there are channels in the cell body that if you have inhibitory neurotransmitters will open those channels, and the chloride ions will flow into the cell body, and because they're negatively charged, they also reduce the membrane potential of the cell. So basically what you have sort of two sources of information, or two sources of ions, two types of ions that are basically reducing the membrane potential of the cell. One, the potassium ions, which are the leak current, that's just sort of a steady state that doesn't respond to anything in particular. The chloride ions, those respond to inhibitory neurotransmitters, so those can change dynamically. The flow of the negative ions into the cell body can change dynamically as a function of the neurotransmitters. And then you have the sodium ions, which are positive ions. And there's what's called a sodium pump, which is continually sort of pumping sodium ions outside of the cell body, which again lowers the membrane potential. But what happens is when you get sort of excitatory neurotransmitters, they open up channels, and then the sodium rushes into the cell body, and because they're positively charged, they're going to raise the membrane potential. Okay, so if you think about this sort of abstract neuron, you've got three different kinds of ions, and you've got different forces moving in different directions. And one way to think about that is then you have this sort of tug of war. And in this particular diagram, for a moment, let's ignore the leak current, which is a constant factor. And you can think of sort of the cell body and the cell and the membrane potential of the cell as this sort of tug of war between sort of the excitatory forces and the inhibitory forces. And the V sub m in the middle of the diagram, it represents the membrane potential, and basically if the excitatory guy can pull harder, the VM or membrane potential moves closer towards him, which is a higher higher charge. Or if the inhibitory guy can pull harder, the membrane potential will move towards him. And the theta there is actually is a threshold, and that'll become important in a little bit, but the idea is if you can get the membrane potential to be higher than the threshold, then the cell will fire a spike. Whereas once the membrane potential is lower than the threshold, the cell will no longer be able to fire a spike. A couple of other things that are important in here is that you can think of an equation I equals G times E minus V sub m, and that's describes how strongly each guy pulls. So G is basically sort of the conductances, or sort of the number of open channels that allow ions to flow into the cell. And so the larger G is, the higher the current is, the higher the effect on membrane potential. And then E and V sub M, basically E is the driving potential, and it's sort of a particular, sort of like a resting state of the neuron, and it's a state towards which the neuron sort of may move in the sort of the absence of other forces. Now, one of the things that's important is that the strength with which the excitatory and inhibitory guy can pull is a function of the difference between V sub M and the driving potential. So basically, the closer V sub M gets to E sub I, the less powerfully the inhibitory guy can pull. Whereas the closer V sub M gets to E sub I, the more powerfully the excitatory guy can potentially pull. And V sub M, which is the membrane potential, reflects the net balance between the two sides. Now, part of what's important is it's not the absolute amount of inhibition versus excitation. It's sort of the relative amount. So you could have you sort of see a resting state where the inhibitory guy can pull a lot stronger. There's a lot more inhibition than there is excitation, so V sub M, the membrane potential, is below the threshold. B is over threshold, where the G sub E guy the excitatory guy can pull a lot stronger. But also, you can have still have the cell over threshold even when you have weaker inputs. So in C, both G sub I and G sub E, the excitatory and the inhibitory guys, are each pulling less strongly, but because they're proportionally pulling less strongly, you still have V sub M above the membrane potential. Above the threshold. So therefore, the cell can still fire. And as I think the textbook pointed out, this is an example of a case in which you can have this sort of dynamic tuning of a cell based on things like background activation. So I think it points out that, like, one of the things that the retina can do is it can control the sensitivity of retinal cells by taking into account background illumination. Now, you can think about this just conceptually, but it is also useful to think about some of the equations that underlie this. Now, if you sort of pull it apart into the equations, you can think of basically what's important is that the current, or the flow of ions into and out of the cell, can be viewed in that top equation as you have the excitatory current, which will be things like the sodium ions moving into the cell, the I sub I, which is the inhibitory current, the chlorine ions moving into the cell, and I sub L, which is the leak current, which is the potassium ions moving out. And then you can sort of calculate the relative importance of those things in terms of the equations. For instance, the first one, you have G sub E times E sub E minus the membrane potential. G sub E is essentially the strength of the conductances, or the proportion of the conductances actually for the excitatory current. So basically, the higher G sub E is, the higher the excitatory current is, the more sodium ions are moving into the cell, and therefore the higher the membrane potential will go. And then the other two parts of the equation, G sub I and G sub L, are similar. So G sub I is again sort of basically the proportion of conductances that are open for inhibitory, the negative potential ions such as the chlorine ions. And so basically then the way you calculate the membrane potential of the cell is basically what was the previous membrane potential of the cell at time t-1 plus the change in the membrane potential, which is basically sort of change in the current. And then you can also pull that apart and combine the first two equations, and then that gives you the bottom equation where you can basically basically look at the membrane potential in terms of previous membrane potential plus the conductances times the differences between the driving potentials for each of the conductances and the membrane potential. And another way to pull that apart and view it is you can think of V sub m as equal to basically in the first term G sub E, which is the excitatory conductances over the sum of the other three, the sum of all three kinds of conductances, so the excitatory, inhibitory, and leak, times the driving potential plus the inhibitory conductances over the sum of the three conductances. So basically you can think of this equation as representing sort of the balance between excitation, inhibition, and leak currents. Okay.